James Howard, Chip Howard to many folks in the audience, clinical neurologist at the University of North Carolina in Chapel Hill, and have in the past life directed the neuromuscular section, run the EMG laboratory. My passion has always been in myasthenia gravis, learned from my mentor during my residency, Dr. T.R. Johns, who was a major figure in that disease. Very active in our clinical trials program, translational research programs, and that's what I focus on over the last several years. Myasthenia gravis is in a revolution, in part because pharma has finally taken an interest in the disease over the last 15 years. And with it, and with all the research that has been done, we are now seeing the first of many newly regulatory approved therapeutics for the disease. In 1955, the FDA Act grandfathered in pyridostigmine, Mestinon, as a treatment, never underwent trial or anything. And so in 2017, by definition, the first regulatory approved treatment for this disease. All of our prior therapies have been borrowed from our transplant colleagues as immune suppressants, and they have been broad in their activity, with the result that, while very effective in many cases, adverse event profiles, etc., which now, because we have choices, become very distasteful to our patients. With all of this, our understanding of the basic science of our new therapeutics has been lagging a bit, and we're in a catch-up mode there. The pipeline is robust. How we practice neuromuscular medicine, MG medicine today, will not be how we're practicing it in 15 years. Zilucoplan is a 15 amino acid macrocyclic peptide. It's not an antibody that targets complement protein 5 as well as protein 5B. In inhibiting the terminal complement pathway, the formation of what in the past was called the membrane attack complex, now the terminal complement complex, which forms a pore-like structure, attaches to the membrane, literally drills a hole through the tissue, leaks its content, and renders it useless. It's what has caused the architectural destruction of the neuromuscular junction. The RAISE trial, which was the blinded portion of the trial, was a 1-to-1 comparison to placebo and demonstrated very rapid and robust efficacy that was sustained through the 12-week point, which was the endpoint of that trial. And then into the RAISE XT trial, which was the open label portion of the trial. We now have data that looks at the 120-week mark, and again have noted very robust, sustained activity. I will say that at the rollover period between the blinded and open label phase, those that were on placebo as well had a very rapid improvement in their outcome measures that then paralleled those who are on active drug from the start. FDA mandates that our primary endpoints are the MG Activities of Daily Living score, a patient-reported outcome measure, but all of our outcome measures that we used in the analyses, from the quantified MG score, the myasthenia gravis composite score, quality of life, even the fatigue score, demonstrated very similar patterns of improvement. As we've gone through the 120 weeks, we have allowed investigators to reduce associated immune therapies. And during this time, we have found that we've been able to reduce those individuals on corticosteroids, to some degree those on other immune suppressive actions. In addition, looking at outcomes, we've seen increasing percentages achieving what we call minimal symptom expression or minimal manifestations. And at 120 weeks, we're approaching some 30% of patients achieving that mark, implying that the longer we treat, we're seeing additional improvement. Why? We clearly don't know. We don't have the science. One hypothesis is that we can actually rebuild the neuromuscular junction, that there's enough plasticity in the nervous system that as we take away this destructive component, that membrane can repair itself. Well, the take-home message is that our targeted therapies work rapidly, unlike any of our other commonly used immune suppressive drugs. They anywhere from 6-12 months to start working, 12 to 36 months for maximum effect. We're talking about onset of action within 1 to 2 weeks, maximum improvement 8 to 20 weeks in time, often on the shorter end of that. We're talking about therapies that have very narrow adverse event profiles, unlike what we see with our other drugs. And we're talking about robustness in the efficacy response. Now, not all patients respond. There's a subset that don't, and we have to find other therapies that do work. But I think that the rapidity of response, the duration of response, or the degree of response, and the sustainability with very narrow adverse events are key hallmarks for our drugs.